I am Christian Tchana, as you said. I am from Cameroon, and I am a PhD candidate at the University of La Rochelle in France. My PhD project will focus on the influence of pesticide use on parasitism and zoonose in farmland specialist bird. Parasitism, like predation, is one of the most widespread ecological interaction in the nature. It is a strong selective pressure shaping population dynamics. In this interaction, the parasite live inside or on the host and gains the benefits. That's why you can see the plus sign for the parasite. And while the host bears the cost, as you can see the minus sign. Parasites are everywhere. Almost every animal species hosts at least one parasite, and parasites may account for nearly a half of living organisms. So, by exploiting their host, parasite can reduce condition, growth, survival, longevity, reproduction, and ultimately lowering host selective value. In host, secondary sexual traits are particularly sensitive to the influence of parasites. A striking example is coloration, which is widely used across many taxa as sexual signals, particularly in mate choice. Colors in animal tissues can result from microscopic structures in the integuments that interact with light. In bird, for example, nanostructures in feather bubbles scatters and reflect light in a specific ways, producing blue or iridescence. So a similar pattern can be observed in moth. Another major source of animal coloration comes from pigmentation. Pigments are molecules that absorb some wavelength of light while and reflect others, and therefore producing colors. Among pigments found in animals, we can have melanins, carotenoids, and other pigments such as pterin, psittacofulvins, and turacoverdin, which are more specific to some taxa. Carotenoids are among the most widely used pigments in animals. And because animal cannot synthesize them, they must be entirely obtained through their diet. And carotenoids are responsible for yellow colors, but can also produce red and orange colors in animals. Carotenoids are not only used in coloration, they are also involved in other physiological functions. They play key roles in immune system and detoxification processes. So this creates a trade-off in carotenoid use. So individuals in poor condition or heavily parasitized may need to allocate most of their carotenoid in immune system and then leaving less available for coloration. So as a result, only high quality individuals may be able to exhibit more intense carotenoid-based coloration. That is why carotenoid-based coloration are coloration are often seen as a honest signals for individual health and parasite resistance. Um, because birds displays many type of coloration, they are often considered as excellent models for studying the influence of parasite on color expression. But most research has focused on endoparasite, especially in relation with carotenoid-based coloration. But how most less is known about how ectoparasite can influence both structural and carotenoid-based coloration in bird. Um, as you may know, nests provide habitat for multiple bird ectoparasites such as malophaga, mites, lice, fly larvae. Among these ectoparasites, uh, fly larvae, especially blowfly larvae of the genus Protocalyphora, are known to parasitize nestlings of altricial birds. Um, they affect more than 100 bird species with a detrimental effect on nestling condition. But they can also induce indirect effect on parents that need to increase their investment in order to compensate for the detrimental effect of ectoparasite on their nestlings. However, uh, uh, the long-term effect of this ectoparasite, meaning the effect of parasite experienced by nestling on yearling coloration has never been investigated. Also, the indirect long-term effect, such as the effect of parent rearing parasitized nestling, has not been investigated. So, given these gaps, our study aim to investigate both the direct and indirect long-term effect of these ectoparasites on different type of coloration in blue teeth. Specifically, we look at how parasites experienced by nestling can influence their coloration once at yearling, and also how rearing parasitized nestling can influence parent coloration. And we predicted that higher parasite load will have a negative impact on nestling condition, on yearling coloration, and parent coloration. Also, we expected this effect to be stronger for carotenoid-based coloration than structural-based coloration. So our study was based on a long-term study, uh, a long-term program of monitoring of blue teeth in Corsica at this population called Pirio, where we can find evergreen forest of oak. Our data have been collected continuously from 2004 to 2023. Blue teeth, as you know, is a small non-migratory passerine distributed across Europe and part of North Africa. It is a dichromatic, sexually dichromatic species as you can see on this map, here is a male because he presents a more brighter blue plumage. During the breeding season, blue tit nest in natural cavities, but can also use nest boxes. This is an advantage. Both parents are involved in feeding chicks, which spend about 21 days before fledge. So for each nest box occupied by a breeding blue-tit pair, uh, we recorded the laying date, the number of eggs, and the number of hatchlings. Also, at 15-day post-hatching, nestlings were wake, their tarsus length were measured, and also these birds were banded with a metal ring with a unique number before fledging. So we use blue feeder and yellow feeder collected at 2 different periods during the nest building and when nestlings were 10 to 15 day old. So for the blue crown, we collect 6 feathers, and for the yellow breast patch, we collect 8 feathers. Feather coloration were measured using a spectrophotometer, and for the yellow breast patch, we measure the chroma, which reflect the amount of carotenoid. We also record the UV chroma and the brightness. For the blue crown, we measure the UV chroma and the brightness. So these 5 color variables are quantitative and continuous and will be used for statistical analysis. In our study population, 2 species of blowfly of Protocalyphora parasites, blue tit nestling. The adult blowfly lay their egg in the nest of blue tit, usually containing eggs or young nestling. And these blowfly eggs, after a few days, develops into blood-feeding larvae, as you can see here, which intermittently feed on nestling, but never feed on fledgling or adults. To estimate our relative parasite load, we count all the parasite life stage and we divide with the number of hatchlings. So this relative parasite load is used for our statistical analysis also. So for our statistical analysis, we first verified if the relative parasite load is a predictor of nestling condition. So we performed 2 linear mix models using respectively nestling wake and tarsus length as response variable. And to test our first prediction, which is the negative impact of parasite experienced by the nestling on their coloration, on yelling, after after the next is first molt. And we build a linear mixed model using all the 5 color variable as response variable. And we use relative parasite load, the number of hatchling, sex, period, laying date as fixed variable. And to test our last prediction, our second prediction, uh, um, to test if parasite, the parasite experienced by nestling have negative impact on parent the following year, study after parent have molt. We also use relative parasite load as fixed variable, number of hatchling, sex, period, age. And we use all our 5 color variable as response variable. We also test for interaction between covariates. So our result concern the relationship between parasite load and nestling condition. We found significant effect of relative parasite load on nestling body mass. As you can see on this graph, we show on the y-axis the nestling mass, on the x-axis the relative parasite load. We can see that the nestling mass decrease as the relative parasite loads increase. However, we didn't find a significant effect of relative parasite load on tarsus length. So this negative effect of the blowfly the blowfly protocalyphora on nestling condition has been previously found in our blue tip population. So this study confirmed this decremental effect of protocalyphora on blue teeth nestling condition. And this can be explained for the high intensity, with the high intensity of blowfly larvae sometimes found in the nest. Here we have the second result, our first prediction, the relationship between the parasite experienced by nestling with their coloration at yearling after their first molt. We can see that our statistical analysis show a significant correlation between the brightness of the yellow breast patch and the interaction between relative parasite load and the number of hatchling. In this graph, you have on the y-axis the brightness of the yellow breast patch, and in the x-axis you have the relative parasite load. You have small brood and large brood. What we can see is that in large brood, the brightness tend to decrease. The brightness of yearling tend to decrease with parasite as the parasite load increase. This suggests a moderate long-term effect on plumage structure. This effect on large brood may indicate that nestling competition can exacerbate the effect of parasite on their coloration at yearling. Anywhere we don't have a strong effect that could suggest possibly a selective disappearance, meaning that maybe some individual that undergone high parasite load die after fledging. So we cannot control for that. For our second prediction, the relation between nestling parasite load and parent coloration, we can see that we have a significant correlation between the UV chroma of the blue crown and the interaction between the relative parasite load and parent sex. And we can see that in female, we have a decrease of the UV chroma of the blue crown as the relative parasite load increase. However, in male, we see that uh, the UV chroma tend to increase. And also, we have a significant relationship between the brightness of the yellow breast patch of the parent uh, with the relative parasite load of their nestling. So this graph shows a decrease of this brightness with the parasite load. So these effects suggest that we have an effect of parasite experienced by nestling on parent coloration, and this uh, this effect also go in the same line with previous study that shown uh, that female contribute more to chick investment than male and may suggest condition dependence of female ornament. But we don't have a strong effect of this ectoparasite on carotenoid-based coloration, uh, which is not online with our prediction. So in conclusion, our study aimed to evaluate the effect of an ectoparasite which only parasites nestling on, on the coloration acquired by, by this nestling and at the yelling stage, and also to look for the indirect effect of nestling parasitism on parent coloration. And what we found is that we have just a moderate long-term effect of this ectoparasite on yearling, and specifically on the yellow breast patch. We also had a moderate long-term effect of this ectoparasite, a moderate and the indirect effect of this ectoparasite on parent ornament. We also find that structural coloration is more affected than carotenoid-based coloration. To investigate more in this area, we can experimentally verify by doing a treatment to see if environmental variable can have a confound effect. We can test it experimentally. We can also take into account both endo and ectoparasite. So all the parasite of individual. Um, I would like to say thank you for everybody. Thank you for the co-author of my study. Thank you for my collaborator, for field team, PhD student and master student. For those who are interested on my work, you can text me, you can send me an email. I can provide you a copy of this work. Thank you so much.
Thank you, Christian. You might like to give a brief French
yeah,
translation. Thank you.
yeah, of course. Bonjour à tout le monde. Je vais faire un petit résumé de ce travail en français. Donc, l'idée de ce travail, c'était de regarder les effets d'un ectoparasite qu'on appelle Protocalifora sur la coloration de la mésange bleue. Et donc, déjà, on est parti du fait que euh, la coloration chez les animaux en général peut être déterminée selon deux modes, soit par la structure du tégument, ce qu'on appelle les couleurs structurales, telles que le bleu, et il y a aussi les colorations à base des pigments, c'est-à-dire la coloration qui est liée à l'incorporation des pigments au niveau du tégument. Donc, et parmi les colorations pigmentaires, on a les colorations à base de mélanine, des, des colorations à base des caroténoïdes. Mais ce qui est pertinent, c'est que les colorations à base de caroténoïdes sont très intéressantes parce que les animaux ne sont pas capables de synthétiser les caroténoïdes. Donc, les caroténoïdes doivent être incorporés par l'alimentation. Et donc, les caroténoïdes aussi, eux, ils interviennent pas uniquement dans la coloration, ils interviennent également dans d'autres processus physiologiques. Donc, les caroténoïdes interviennent par exemple dans le système immunitaire, la stimulation du système immunitaire et le phénomène de détoxification. Donc, ce qui fait que euh, les colorations à base de caroténoïdes sont soumises à ce qu'on appelle un compromis. Donc, si un individu est malade, cela signifie qu'il va utiliser ces caroténoïdes pour le système immunitaire, pour lutter contre le parasite. Donc, en écologie, il est généralement connu que euh, les individus parasités auront des colorations moins exubérantes, c'est-à-dire des colorations moins fortes, alors que les individus qui ne sont pas parasités, ils utilisent toutes leurs caroténoïdes pour leur coloration, donc ils auront une coloration plus brillante. Donc, la coloration est donc, permet donc de définir ou bien de prédire si un animal est en bonne santé ou pas. Donc, partant de là, euh, il existe un ectoparasite qui est connu pour parasiter uniquement les poussins. Et ce parasite s'appelle Protocalifora. Ce sont des larves de mouches. Et ces larves de mouches, n'est-ce pas, c'est-à-dire on a des mouches adultes qui vont pondre les œufs dans les nids et ces œufs vont donner des larves et ces larves vont se nourrir exclusivement sur les poussins et pas sur les adultes. Et donc, les études ont montré que ces larves de mouches-là, qui sont hématophages, n'est-ce pas, entraînent des effets négatifs sur la condition des poussins. C'est-à-dire, les poussins ont par exemple une diminution de leur masse corporelle, une diminution de la longueur du tarse et aussi une diminution de l'hématocrite. Et donc, nous, on s'est dit, si les poussins sont dans des mauvaises conditions à cause du parasitisme, est-ce que cela ne pourrait pas avoir à long terme des effets négatifs sur leur coloration? Sachant que la coloration est censée être un signal condition dépendant, c'est-à-dire que faible condition égale faible coloration. Et donc, pour tester, on a aussi essayé de regarder parce que lorsque les poussins sont parasités, les études ont également montré que les parents augmentent leur investissement. Par exemple, en nourrissant des poussins de plus en plus. Donc, les parents sont aussi affectés par le parasitisme de leurs poussins. Et donc, on s'est aussi donné comme hypothèse de tester si le fait que les parents soient très en activité lorsque les poussins sont parasités, est-ce que cela n'affecte pas leur coloration l'année d'après? Et donc, nous avons donc, pour tester ces hypothèses, nous avons fait des modèles linéaires. On a euh, testé la corrélation déjà entre la charge parasitaire des poussins et leur masse corporelle. Et ensuite, nous avons testé la corrélation entre la charge parasitaire des poussins et leur coloration l'année suivante, c'est-à-dire après leur premier mu. Et enfin, on a testé la corrélation entre la charge parasitaire des poussins et la coloration des parents l'année d'après également. Et donc, les résultats ont montré que, pour la première question, que, en fait, plus la charge parasitaire augmente, plus les poussins perdent de poids. Donc, ce parasite diminue le poids des poussins et donc diminue la condition des poussins. Et maintenant, pour la deuxième question, nous avons remarqué que, en fait, l'augmentation de la charge parasitaire entraîne une diminution de la brillance du plumage jaune chez les jeunes, c'est-à-dire les jeunes qui ont subi le parasitisme une fois qu'ils sont, après leur première année, une fois que l'année d'après, ils ont la brillance de leur plumage diminue. Et en ce qui concerne les parents, on s'est rendu compte également que lorsqu'ils ont des poussins parasités dans leur nid, euh, la coloration bleue, leur coloration bleue, qui est une coloration structurale, elle diminue également. Mais cette diminution est plutôt marquée chez les femelles, alors que chez les mâles, on n'observe pas cette diminution. Et en ce qui concerne le plumage jaune, on observe également une diminution de la brillance. Donc, cette étude a été faite, je ne l'ai pas précisé, chez la mésange bleue, parce que la mésange bleue porte deux types de coloration. Une coloration jaune qui est à base de caroténoïdes et une coloration bleue qui est une coloration plutôt structurale. Donc, voilà l'essentiel de ce travail.